Hello everyone, welcome to Agri Addict YouTube channel. I hope you all are doing well. So my name is Sakhi. Today I'll be discussing the photosynthesis in plants lecture 2, part 2. This is the crop physiology lecture series. So let's move. This is my short intro. I have done my undergraduation from JNKV Jabalpur. Currently pursuing masters at AIR Hyderabad Hub FREDA, Department Agronomy. I have secured All India Rank 12th in ICR PG exam 2024. So before starting this lecture, what we have discussed earlier in the photosynthesis part 1 is the all the experiment. But first we discussed what is photosynthesis, and then we discussed the different experiments by which we got to know about photosynthesis, like what is important, engine house experiment, like Presley's experiment, several experiments we studied in the previous lecture. And then we studied about the pigments, their arrangement, and how they're trapping light, how they're converting into chemical energy. So that we studied in the previous lecture. In this lecture, we'll get into the mechanism of photosynthesis. Okay, so let's first start with the first part comes is the light reaction. Okay, light reaction. Why we are saying it light reaction? Because it is directly dependent on light. Here, light is directly required. This produces energy, and site of light reaction is thylakoid membrane. In the previous lecture, we studied the structure of chloroplast, right? In that I have showed you, might be remembering this. This is the chloroplast per se. It is a double membrane structure. Okay. In that we have studied, wait a minute, this coin-like structure. This is what the thylakoid is. And when we are studying the stack of thylakoids, then this we call it as grana. So for the site, when we are concerned about site of light reaction, so this thylakoid membrane is the site of reaction. So here I have enlarged it. So per se, this is one thylakoid. So in this membrane, that chlorophyll pigment will be there, which will help in trapping the incoming sunlight. So here this light reaction will be taking place. So you should visualize it where the process is actually taking place. So it will help you to remember better. Okay. Now comes so what happens in light reaction is here light energy is getting converted into chemical energy. Okay. And this chemical energy which is being produced here, what chemical energy will be produced will be ATP and NADPH2. This is the energy which will be produced in light reaction, which will be utilized in dark reaction for sugar synthesis. Sugar synthesis means glucose synthesis. Okay. So this is how the light reaction is. This is a basic intro of light reaction. Now let's come to the in detail light reaction. Light reaction is divided into two. Like first is non-cyclic photophosphorylation and second is cyclic photophosphorylation. When we are telling phosphorylation, it means there is the formation of energy. And here we are saying photophosphorylation means formation of energy or synthesis of energy in presence of light. So photophosphorylation. So first is non-cyclic photophosphorylation and second is cyclic photophosphor. Why we are calling it one as non-cyclic and another as cyclic, we'll understand in the coming slides. So let's move. So first is non-cyclic photophosphorylation. So this reaction is all this process is also known as Bendel and Hill reaction. Okay, this you have to remember. And site for this non-cyclic photophosphorylation is oppressed region of thylakoid membrane. What is oppressed region? For say, this is the thylakoid stacks. Okay, this is inside this chloroplast. Chloroplast is the double membrane structure. Okay, so this is the thylakoid. Now the part of thylakoid which is in contact with another thylakoid, this is called oppressed part. In the previous lecture also I have taught this. This, the membrane which is connected to thylakoid, so it is not in direct contact with the stroma liquid, which is the fluid inside the chloroplast. Okay. So this part is known as this oppressed region. Okay. And the part of thylakoid which is in direct contact with stroma, this portion which is in contact with stroma, this is known as unoppressed part. Unoppressed part or unoppressed region of thylakoid membrane. So non-cyclic photophosphorylation takes place in oppressed region of thylakoid membrane. So we can say that it occurs in grana only. Because in the previous and where I taught you about the structure of this thylakoid, in that I also told about the stroma lamellae. Stroma lamellae is the structure which connects one grana to the another granule structure. So here this non-cyclic will not occur in this stroma lamellae. It will occur in only the granule thylakoid like this, where this oppressed part, in that too in oppressed part. Okay, so you do not have to get confused like where it is because I am explaining you thoroughly like what is oppressed part, what is unoppressed part, so you do not have to confused in that. So that's why we are telling here occurs, it occurs in grana only. Now comes the requirement. What are the requirements? First, we'll require PS1, PS2. In the previous lecture, we studied what is PS1, that is photosystem 1. Now, second is PS2, which is photosystem 2. Third is ATP synthase molecule, and then NADP reductase. These two enzymes we need, and these two photosystem is required, PS1 and PS2. Now let's come to the next slide. Here are some abbreviations given. In the next slide, I have included a cyclic representation of it, non-cyclic photophosphorylation. So this is how the non-cyclic phosphorylation takes place. It is also known as Z scheme. Why we are saying it is known as Z scheme? If you will see it as C, this is how the structure is. The energy flow. First, it is uphill, then downhill, then uphill. So that's why it is known as Z scheme. Okay. Now, first, let's start with the. So, this is the outer membrane. This is the outer membrane of chloroplast. And this is the inner membrane of chloroplast. Okay. This you have to remember. Inside this inner membrane, this is lumen. Okay. Let me change the color. Okay. This is lumen, and this outer membrane outside of it is stroma. Here the pH is 7 to 8. Okay, in stroma. And in lumen, the pH is generally between 2 to 3. Why we'll study it in the further session. Okay. Now, first what we'll do, this is the PS2, which is photosystem 2, and this is photosystem 1. So, we studied in the previous lecture that photosystem 2 absorbs energy of 680 nanometer. Okay. So when a 680 nanometer wavelength of light will fall on PS2, what will happen? PS2 is made up of, in the previous class, what we studied is, this is how the chlorophyll is arranged. Okay. This is for say chlorophyll A, we call it as reaction center. And around to it are the other accessory pigments. Okay. So this we are calling as PS2. Okay. This PS2 will take energy of 680 nanometer. And this, as we know, this chlorophyll have magnesium at the center. So what will happen when energy will fall on it? This magnesium will release an electron and will become magnesium plus. Okay. So here what happens in PS2, this magnesium will release because it is getting energy. This the structure will get like the magnesium will release energy, will get energy and will release electron. Excited that electron will get excited. So this excited electron will be first accepted by this PO phytin. This is important. Okay. Now this electron will be transferred from phaeophytin to plastoquinone. So in this place, in this plastoquinone, what happens? This plastoquinone will take this electron. While taking this electron, this plastoquinone, what it will do? It will take H plus ion from stroma. And then this plastoquinone will get converted into reduced form of plastoquinone. Okay. And now what it will do during the transfer of this electron to cytochrome B6F complex, it will also transfer an H plus ion from stroma, and this will it will put into the lumen. Okay, so every time the electron is getting released from this magnesium, this PS2, so every time this H plus will be coming from this. Just like you can understand it as like for say we are eating samosa. Okay, so chutney is a must with samosa. So that's why here if you can like make it a analogy like that. So if and here the electron is the samosa for plastoquinone, and now if samosa is there, so plastoquinone needs chutney also. So what he'll do, it'll take one H plus from stroma, and then it will transfer this H plus to the lumen, and then it will pass on the electron to cytochrome B6F. I just use it as samosa chutney. You do not just to remember like while transferring electron, it is also taking one H plus from stroma and it passing it on to lumen. So what happened next is this electron is getting transferred to cytochrome B6F complex. Now this electron will get transferred to plastocyanin. This PC is plastocyanin. Then plastocyanin will take this electron to PS1. Now what will happen? This electron is coming to PS1 and this 700 nanometer light is falling on PS1. Okay. Now what happened? Here also magnesium is there. This magnesium will release its outer shell electron. So this electron will get excited and this electron will be accepted by this iron sulfur protein. Then this iron sulfur protein will transfer the electron to ferredoxin, and now this ferredoxin will transfer it to FNR, ferredoxin nitrate, NADP, ferredoxin NADP reductase. Okay, this FNR complex, what it will do, it will transfer the electron to the NADP plus. This NADP plus and H plus are coming from stroma. It will transfer the electron to this, then what will happen? There will be a formation of NADPH or NADPH2, or we can call it as NADPH plus H plus. These all the three are similar. We can represent it by different ways. The molecule is same, NADPH2 or NADPH or NADPH plus H plus. So at the end, what is happening? The electron which was released from PS2 is getting is accepted by this NADP plus. So here what we are saying is this NADP plus is the final electron acceptor. Okay. This much I hope you have got. What is happening is in PS2 this 680 nanometer light is falling. This magnesium is releasing its outer electron. This electron is getting excited. This electron is getting accepted by phaeophytin molecule. Okay. This phaeophytin will transfer it to plastoquinone. While transferring electron to cytochrome B6F, this plastoquinone, what it will do is it will take H plus from stroma and transfer it to this lumen. Okay. Now this electron, the cytochrome B6F complex will transfer the electron to plastocyanin. Now this plastocyanin will transfer the electron to PS1. Now, at the same time, the sunlight, 700 nanometer wavelength sunlight is falling on PS1. Here also the magnesium will release its outer electron. This electron is getting excited. This electron is accepted by iron sulfur protein. Then it transferred to ferredoxin. Now transferred to ferredoxin NADP reductase. And this NADP reductase will transfer this electron to NADP plus H plus. This is from stroma and it will get converted into NADPH or NADPH2 or NADPH plus H plus. Okay, this is the scenario in non-cyclic photophosphorylation. Now the question comes here, this magnesium is releasing one electron. Now this is magnesium 1 plus. Now this magnesium again it has to excite one electron. So we have to fulfill the requirement of this magnesium. So how it will be filled? So here this oxygen evolving complex is there, OEC. This is I have written oxygen evolving complex. What are the elements which are there? It is manganese 2 plus, chloride ion and calcium ion. This is the part of oxygen evolving complex. So this oxygen evolving complex, what it will do, it is present in the inner membrane. Okay, this is the inner membrane. Here it is present oxygen evolving complex. It will take H2O, break it down into 2H plus, 2 electron and half of O2. Okay, this O2 will be evolved in air. Okay, now this H plus is in lumen. One H plus is coming every time the electron is getting transferred. H plus is coming into the lumen. Now this H2O is broken up by this oxygen evolving complex. Here also the H plus is getting added. That's why we are saying the lumen pH is 2 to 3. Why? Because H plus is coming. When the concentration of H plus ion is increasing in the lumen, the pH will decrease. Okay. Now the requirement of this magnesium for electron, this will be fulfilled by this 2 electron. Each H2O molecule, when it is getting broke up, then it is releasing 2 electrons. So it is fulfilling the requirement of magnesium. For say this energy is falling, the electron went out. Now this electron will get replenished. This magnesium will get another n electron and then it will become magnesium. Then again the light will fall. It is getting excited. So this is how the continuity of this electrons transport is maintained by this splitting of H2O, where who is responsible for it? It is oxygen evolving complex. Okay. Now, this photocyanin, so this plastocyanin, connecting link between PS1 and PS2 in non-cyclic ETC. This. Okay. So this you have to remember what is the connecting link between. So that is plastocyanin. Hmm. Now comes the chemiosmosis. This you have understood how it is the electron is getting transferred, how the NADPH2 is formed. So this you remember. Okay. Now comes this chemiosmosis. So as this H plus is coming from stroma, then for the splitting of H2O also increasing the H plus concentration in the lumen. So what will happen is the chemoosmosis process, there is one this ATP synthase enzyme. Okay, it is having CF naught unit and CF1. Two units are there. So CF naught unit, it is longitudinal in structure and the CF1 is round shape in structure. So here it is arranged in between this membrane. This is the inner membrane, it is the outer membrane. In between the CF naught molecule is there, and outer side of this membrane, in stroma, this CF1 molecule is there. This is the ATP synthase enzyme. Okay. So first molecule we have formed. We studied in the previous slide that light reaction we get two chemical energy we get. First is NADPH2, second is ATP. So NADPH2, how it is formed, we studied. Now we have to study how ATP is formed. So what will happen? Because this H plus is getting transferred from stroma to lumen of thylakoid membrane, thylakoids. So what will happen? There will be a potential gradient. This H plus concentration gradient will be there. Here H plus is more, here H plus is less. So what will happen in this during this chemiosmosis? What will happen? This H plus will get transferred from this molecule to outside to stroma to maintain this whatever the potential is concentration gradient is there, this H plus will get outside through this ATP synthase molecule, and during this there is this is kind of rotatory structure. Okay, so here in from inside the H plus it will take this enzyme will take H plus, and this the it will rotate like a machine. Okay, then it will rotate and remove it through the in this stroma. So during that, what will happen from stroma, ADP molecule plus inorganic phosphate molecule, this will combine and will form the ATP. This is what the chemiosmosis is during chemiosmosis, how ATP is formed. This H plus which is present inside the lumen will get out through ATP synthase molecule. This ATP synthase molecule will turn like this and will transfer this H plus from lumen to stroma. During that time, what will happen? This ATP synthase molecule will take ADP from stroma, inorganic phosphate from stroma. It will combine the two and form the adenosine triphosphate molecule, which is known as energy currency. Okay. So for this chemiosmosis to take place, this concentration gradient of H plus is required. When H plus will go out, then only this ATP synthase will be able to form this ATP. Okay, I hope this you have understood. Okay, so here one note is there that H2O is electron and hydrogen donor. This we have already studied. Here H2O is getting splitted in presence of light. So H plus, 2H plus, 2 electron and half O2. This is releasing into the air. Now, primary electron acceptor is pheophytin. Sometimes what happen in questions, this pheophytin option is not given. If pheophytin option is given, and then the primary electron acceptor is pheophytin. Okay. If this option is not given, then plastoquinone you have to tick if pheophytin is not there. Okay, if pheophytin is there, you go for pheophytin. It's the correct answer. But if pheophytin is not given in like all the four options, pheophytin is not there, then go for plastoquinone. Okay, let's move to the next slide. Now the explanatory. Both PS1 and PS2 are involved in non-cyclic photophosphorylation. In the previous we studied here PS2 and PS1 both are involved in non-cyclic photophosphorylation. Now, so it occurs in at grana thylakoids. I already told that it occurs in grana thylakoids because PS2 is present in here I dot. So in grana thylakoids only it will take place. Okay, not in stroma lamella because in stroma lamella this PS2 photosystem is not there. Not in stroma thylakoids because stroma thylakoids lack PS2 as well as NADP reductase enzyme. This NADP reductase enzyme is also not there in stroma thylakoid. That's why this non-cyclic photophosphorylation takes place in grana thylakoids only. Now comes the primary electron acceptor from PS2 is pheophytin. This we already studied, which passes electron to an electron transport system. That what we studies as Z scheme. Okay, consisting of cytochrome. There are different uh, protein molecules. Like the cytochrome is a transmembrane protein. In the previous here it is written. See, this is a transmembrane protein. It is connecting the inner membrane and the outer membrane. So that's why it is known as transmembrane complex. This is the electron transporting chain. These proteins are what they are doing is they are transferring electron. They are passing electron from one protein molecule to another, which passes electron to the electron transport system consisting of cytochrome. This movement of electron is downhill, okay, in terms of an oxidation reduction or redox potential. The electrons emitted from PS2 pass through the electron transport chain to the pigments of PS1. This is how the electron is getting transferred. First electron is released from PS2, it is getting transferred through electron transporting chain, and now it comes to PS1. Okay, this is what here it is telling. Simultaneously, electrons in the reaction center of PS1 is also excited because when sunlight is falling, it will fall on the PS2 also, PS1 also at the same time. Okay, so PS2 is requiring 680 nanometer wavelength light. So this much wavelength is required to emit its electron. At the same time, this PS1 is also emitting its electron because PS2 is getting this electron from this splitting of water. So this so that the cycle can get continue. So the electron which is getting released from PS2, it will fulfill the requirement of PS1 because at PS1 we do not have any photo splitting of water. So once the electron from magnesium is getting released to fulfill that electron need, this photosystem 2 is fulfilling that electron need of photosystem 1. Okay, I hope you got what I wanted to say. Now, simultaneously, electrons from reaction center of PS1 are also getting excited when they receive red light wavelength of 700 nanometer and are transferred to another acceptor molecule, that is FRS, and then to paradox. Okay, iris alpha protein, then paradoxin, and then we had FNR, which is paradoxin NADP reductase. Next, these electrons will then move downhill again, and this time to a molecule of energy-rich NADP plus. And this NADP plus is coming from stroma. It is getting combined with H plus, and it is forming NADPH2 molecule, which is an energy, chemical energy, which is getting produced in this non-cyclic photophosphorylation and light reaction. Next. Now we have to know what are the products which is getting formed in non-cyclic photophosphorylation. First, due to photo splitting of water, this O2 is forming due to photo splitting of water. And this ATP is formed via chemiosmosis. Okay, chemiosmosis. This NADPH2 is formed through this electron transport chain. Now each cycle produces one ATP and one NADPH. Okay. If four molecule of H2O, it is okay, yeah, four molecule. Four molecule of H2O, when the photolysis of water will take place, four more, then 1 mole of O2 will be released. Okay. So let's move to the next. Now comes the cyclic photophosphorylation. In the previous, we studied non-cyclic. Why? Because once the electron is getting released, it is not coming back to PS2. Once electron is getting released, this electron is getting transferred to PS1. This electron is getting transferred to this NADP, and NADPS2 is being created. No, this electron which is getting released from this PS2 is not coming back to this PS2 again. So that's why we are saying this is a non-cyclic photophosphorylation. It is the energy formation of process in presence of light where the electron is not getting cycled back to PS2. That's why we are calling it as non-cyclic photophosphorylation. Now comes the cyclic photophosphorylation. Why we are saying it's cyclic, we'll understand in the next slide. So first we have to remember is the site of cyclic photophosphorylation. It is the unuppressed region of thylakoid membrane. Unuppressed region means the regions which are in direct contact with stroma, that fluid. Okay. So, wait, here. This portion which is in direct contact with stroma, the stroma lamellae portion also, it is in direct contact with stroma. So here it is taking place. The cyclic photophosphorylation is taking place in unuppressed region of thylakoid membrane. So we can say that it occurs in both grana and stroma, because in previous we studied non-cyclic photophosphorylation. In that we studied that it is occurring only in grana. Here it is occurring in grana thylakoids also and stroma thylakoids also. Here, no O2 evolution is taking place. The difference between cyclic and non-cyclic. Okay, in non-cyclic, we studied oxygen is getting released because there is photolysis of H2O. Photosplitting of H2O is taking place of water, and which is responsible for the release of O2. Here, this H2O photolysis will not take place in cyclic photophosphorylation. We'll study how. And no NADPH2 is formed. Here NADPH2 is also not formed. Here 2 ATP is generated in this cycle. Okay, 2 ATP molecules are being generated. No NADPH2, no O2 evolution. This is the difference, basic difference between non-cyclic and cyclic. Now comes the requirement. Okay, so requirements here, only PS1 is required. There PS1 and PS2 both were required. Here ATP synthase molecule, synthase enzyme is there. There ATP synthase was also there. There NADP reductase was also there. PS1, PS2, four things were required. Here only PS1 and ATP synthase. Okay, two requirements are there. Now comes the cyclic photophosphorylation. It is very easy, not complex as non-cyclic. So here what is happening? Sunlight is falling. This 700 nanometer is falling on PS2. This magnesium is there. When sunlight, the energy will fall on it, what will happen? The electron will get excited. This electron will be excited. And this excited electron will be accepted by this iron sulfur protein. It is the first electron acceptor. Paradoxin reducing substance also we call it. Paradoxin reducing substance. Now this electron is getting transferred from iron sulfur protein to paradoxin, and this paradoxin will transfer this electron to plastoquinone. In the previous also, what this plastoquinone needed is, it needs samosa with chutney. So when electron is getting transferred, for say this electron, we are saying it as samosa. So if it is getting electron, so it needs chutney also. Chutney is this H plus, which is coming from stroma. So what it will do while transferring this electron, it will transfer this H plus again to the lumen. Okay. Now this plastoquinone will transfer this electron to cytochrome B6F complex, which is a transmembrane protein. Then it is getting transferred to plastocyanin. This plastocyanin will transfer the electron back to PS1. That's why we are calling it as we are calling it as cyclic photophosphorylation, because here the electron which is released from this PS1 is getting again back to this PS1. That's why here no H2O splitting is there, because no electron source is there. So that's why this electron has to come back to PS1. So that's why we are telling it that it is cyclic photophosphorylation. Okay. Here also, because of this H plus transfer, the lumen pH will be less. Okay. Lumen pH will be less, 2, 3, because more H plus is there inside, and compared to stroma, here the pH will be somewhat neutral. So this chemiosmosis is taking place. Now this ATP synthase, this is the ATP synthase molecule. This ATP synthase molecule will transfer this H plus to the stroma back. While transferring this H plus, what it will do, it will take this ADP molecule, adenosine diphosphate plus inorganic phosphate, and will convert it into ATP. So here only one energy molecule is getting produced, that is the product of cyclic photophosphorylation is only ATP. No oxygen is released, no NADPH2 is formed. Okay. So this is simple, very simple compared to non-cyclic. Now, in cyclic photophosphorylation, only PS1, only PS1 works. Okay. Possible location for this could be this stroma lamellae or stroma thylakoid, although this is also occurring in grana thylakoid, but in the unuppressed part, which is in direct contact with the stroma. During cyclic electron transport system, the electron ejected from reaction center of PS1 returns back to its reaction center. At every step, oxygen and reduction of each electron carrier takes place, which releases the energy, and this energy is utilized in creation of proton gradient, this H plus gradient which I talked about to form ATP. The chemiosmosis will take place. The H plus, this ATP synthase, it will take H plus out. While taking out the H plus, it will rotate. The machinery will rotate. It will release the H plus. Then the ADP plus inorganic phosphate from stroma will combine and form the adenosine triphosphate, that is energy currency of the cell. In cyclic ETS, no oxygen evolution occurs. In the previous slide, I told because photolysis of water is absent. There is no photolysis. Okay. NADP H plus H plus, this is reducing power, is not formed in cyclic process. So there is only the formation of ATP. I hope you have understand till the time. So let's move to the next. Now that chemiosmosis we studied there, this concept was given by Peter Michel. And what are the requirements here? The membrane is required, a proton pump is required, ATP synthase enzyme is required, this proton motive force or proton gradient is required. The concentration difference between H plus ion which is present in stroma and the H plus ion which is present in lumen. So why the proton gradient is being formed? Because this NADPH2 formation is there. What is happening is this NADP from stroma is combining with H plus. So what will happen? It will form NADPH2. So H2 is getting used to produce this. So their H plus concentration is decreasing by this mechanism. Now this proton pump is there. What is this proton pump? This plastoquinone is transferring. The chutney and samosa concept you remember. Okay, while transferring electron, it is taking H plus ion from stroma and transferring it to lumen. And then the third reason for this proton gradient is photolysis of H2O. This oxygen evolving complex is there in case of non-cyclic. Okay, there inside the inner membrane of thylakoid, it is present there. So here the H2O is getting splitted, and in that case the two H plus ions are released. So the concentration of H plus inside the lumen is getting increased. So this is responsible for this proton gradient. Now comes the basic differences between non-cyclic photophosphorylation and cyclic photophosphorylation. Both PS1 and PS2 are involved in non-cyclic both the photosystems are involved in non-cyclic. We studied already. Only PS1 is involved in cyclic. We have seen only PS1 is working for cyclic process. First electron acceptor from PS2 is pheophytin, very important. And here first electron accepted from PS1 is FRS. Okay. The electron expelled from reaction center is not cycled back. It is not coming back to PS2. Okay. Its loss is compensated by electron from H2O because photolysis of water is taking place. So the requirement of electron is getting fulfilled by that. Here, the electron expelled from P700, that is PS1, is cycled back. We are getting again that electron is getting supplied to the PS1 to fulfill the configuration of magnesium. Okay. Photolysis of water and evolution of O2 takes place. Here, photolysis is taking place. Because of that, O2 is getting involved. Here NADP plus is reduced to NADPH2. Here it is not the case. NADP is not reduced. Here photolysis of water is not taking place. Evolution of O2 is not taking place. This is the basic difference between non-cyclic photophosphorylation and cyclic photophosphorylation. Let's move to the next slide. Now comes the dark reaction. We have completed light reaction. In light reaction, what is happening? This light energy is getting converted into chemical energy. What is chemical energy? This is ATP, NADPH2. Okay. Now the light reaction has done its work, and now this ATP and NADPH2 will be used in the next step, that is dark reaction. Why we are calling it dark reaction is, it is not like that it will occur in night only. No, this concept you have to remember, that it is calling we are calling it dark reaction because it is not directly dependent on light. Okay, in case of light reaction it is directly dependent on light, that's why we are calling it as light reaction. But in case of dark reaction, it is not directly dependent on light. Okay, so it cannot take place in dark. Why? Because it requires this ATP and NADPH2. If light will not be there, so light reaction will not take place. So we cannot say that we are calling it dark reaction because it can take place in dark. No, because it is requiring the products of light reaction. So we cannot say that it cannot take place in. So we'll say that it cannot take place in dark. Okay. Indirectly dependent on light, because light reaction, the products of light reaction are being used in dark reaction. It is a light temperature, here light and temperature required for enzyme functioning. Okay. So a dark reaction, we are just saying because it is not directly dependent on light, but it requires light because it requires product of light reaction. So here the site is stroma. Light reaction we studied, it is the granule thylakoids and stroma lamellii. Here the dark reaction takes place in stroma. It is also known as biosynthetic phase. In this process, CO2 is reduced to sugar. Which sugar? Glucose. The 6 carbon sugar or a hexose sugar. It is known as dark reaction, but it doesn't mean that it occurs in dark. The same concept. Dark reaction we are calling it, but it doesn't mean that it will take place in night only. This is not true. This process does not uh, This process does not directly depend on the presence of light, but it is dependent on the products of light reaction. Not directly dependent on light, but depend on the products of light reaction. So light is important because if the light will be there, then only the light reaction will take place, then only the ATP and NADPH2 will be synthesized. So ATP and NADPH2 is the product of light reaction. So immediately after light becomes unavailable, the biosynthetic process continues for some time and then stop. When light is not there, this light reaction will get stopped. ATP and NADPH2 will not get produced. It will not be able to come. That dark reaction will not take place because ATP and NADPH2 is not there. If then light is made available, after some time, if light is made available, the synthesis will start again. Now, the dark reaction is divided into 3 pathways based on the type of plant in which it is taking place. So, first is C3 pathway. C3 pathway is taking place in C3 and C4 plants, whereas C4 pathway is taking place in C4 plants. Okay, C3 plants means like rice, wheat. Okay, so these are C3 plants. C4 plants, maize, sorghum. Okay, like this. And CAM pathway. CAM pathway takes place in CAM plants. Crustacean acid metabolism plants. CAM plants. Like pineapple, agave. Ah, sisal. Sisal, okay. Pineapple and sisal, these are CAM plants. Next comes the Calvin cycle or C3 cycle. So this Calvin cycle was given by Melvin Calvin, the scientist, very famous scientist. Here the site of Calvin cycle is stroma of chloroplast. This you have to remember. These kind of question will be expected in the JR examination. Stroma of chloroplast. So this is seen in all plants. Three main steps are there. Calvin cycle in simple form we can divide it into three steps: carboxylation, reduction, and regeneration. Okay, so it is actually a very complex cycle, but to our understanding, to our easiness, it is divided into three parts. First is carboxylation, second is reduction, third is regeneration. So what will happen here? This RUBP is there. Wait. Hmm. This RUBP is there. What is this RUBP? It is ribulose 1,5-bisphosphate. It is a 5 carbon compound. Okay, this RUBP. So this RUBP, it will take carbon dioxide from atmosphere in presence of this enzyme rubisco. What is this rubisco? It is ribulase 1,5-bisphosphate carboxylase oxygenase. Carboxylase means it is able to fix carbon dioxide, and oxygenase means it is also able to fix oxygen. Based on the concentration of carbon dioxide or oxygen in the cell, this rubisco will function. Okay, so this rubisco, what it will do when the carboxylase part is function, like carbon dioxide concentration is more in the cell, in the chloroplast, then what will happen? This RUBP will combine with its CO2. Okay, this carboxylation will take place in presence of rubisco. Then the two molecules of 3-phosphoglyceric acid will be formed, or 3-PGA will be formed. It is a 3-carbon compound. Because the first product which we are getting is a 3-carbon compound, we are calling it as C3 cycle. The reason why we are calling Calvin cycle as C3 cycle is the first stable compound which we are getting is a 3-carbon compound. Now what is happening? This is the first process, carboxylation. Now this 3-PGA or 3-phosphoglyceric acid here, 2 ATP and 2 NADPH2 is getting combined, or then what is happening? It is getting converted into 2 ADP plus 2 inorganic phosphate plus 2 NADP. Now this 3-PGA, when we are involving this 2 ATP and 2 NADPH2, it is getting converted into triose phosphate. What it is? It is two molecules of 3-phosphoglyceraldehyde or 3-PGAL. It is also a 3-carbon compound. Here reduction is taking place. Earlier it was acid, means COOH, carboxylic acid. Now it is CHO. Oxygen is getting released. So here reduction is taking place. This is the aldehyde molecule. Here from RUBP 5 carbon, 1 CO2 is getting compound. It is 6 carbon. So now two molecules of 3 carbon compound is formed. 3 to the 6. Okay, balanced. Then again this two molecule is getting reduced to 3-phosphoglyceraldehyde. Now these two molecule are there. One molecule will go for glucose formation and one molecule will go for regeneration of this RUBP. Because to fix this carbon dioxide, we need this RUBP. To generate this RUBP again, the third process is there. It is regeneration. Here again one ATP molecule is required. It is getting converted into ADP and inorganic phosphate. Now what is happening is this 3-PGAL, one molecule which is left is converted into RUBP, a 5-carbon compound. This is the whole Calvin cycle in brief. First is carboxylation, second is reduction, third is regeneration. Now this is also known as C3 cycle. Why? Because the first stable compound formed is a 3-carbon compound. Okay. This rubisco enzyme is activated by magnesium and red light. This you have to remember. This is Calvin cycle or C3 cycle. This is taking place in stroma of chloroplast given by Melvin Calvin. Okay. Next. To fix one carbon dioxide, 3 ATP and 2 NADPH2 is used. How? See, 2 ATP is used here and 1 ATP is used here. How many ATPs? 3 ATP. Okay, 3 ATP is used. Now, previous here, 2 NADPH2 is used. So, to fix one carbon dioxide, 3 ATP is used and 2 NADPH2 used. One cycle is taking place to fix only one carbon dioxide. To fix one carbon dioxide, 3 ATP is required, 2 NADPH2 is required. Now, for one glucose, if we have to form one glucose, it is a 6 carbon compound, means 6 Kelvin cycle is required. 6 carbon dioxide fixation should be there. So for that, we need 6 Kelvin cycle. If 1 Kelvin cycle requires 3 ATP, then 6 Kelvin cycle will require 3, 3 into 6, that is 18 ATP. Because 6 times that cycle will run, then only the one glucose will form. Similarly, if one cycle is requiring 2 NADPH2, so 6 cycle will require 12 NADPH2. This you have to remember. For formation of one glucose, 6 CO2 fixation is required. To fix 6 carbon dioxide, the Calvin cycle has to be run 6 times. So for that, we need 18 ATP and 12 NADPH2. And this ATP and NADPH2, from where it is coming? It is coming from light reaction. Okay, this is the Calvin cycle. Now comes the photorespiration. See, this Rubisco has two parts, carboxylase or oxygenase. When the concentration of carbon dioxide in cell is more, then this C3 cycle will take place. For when O2 concentration is high, then this enzyme will work as oxygenase. So what is happening is this photorespiration will take place in that time. So it is a wasteful process. Why we are telling? Because there is no carbon dioxide fixation, no net energy is produced, fixed carbon dioxide is released. Whatever the carbon dioxide plant is fixing, in this process, this carbon dioxide is also getting released. It is completely a waste process. And it is also utilizing one ATP which is produced in light reaction. It is also known as C2 cycle or photosynthetic carbon oxidation cycle. Important, important. This C2 cycle is important. Now, here this photorespiration requires 3 organelles. First is chloroplast, double membrane structure. Second is peroxisome, single membrane. And third is mitochondria, double membrane. 3 organelles are required. Organelles. First is chloroplast, peroxisome, and mitochondria. Next. Now this is the cycle. So first we talked that chloroplast is required, peroxisome, and mitochondria. So here what is happening? This RUBP, that RUBP we studied in Calvin cycle. Here, this RUBP, ribulose 1,5-bisphosphate. This here also it is fixing this oxygen because the concentration of oxygen is more in cell. Now this Rubisco oxygenase part will work. This oxygen will be fixed. Then it will get converted into 2-PGA. Okay, with the 3 carbon compound, this will go into Calvin cycle. Now there another molecule is formed is phosphoglycolate. This phosphoglycolate is a 2 carbon compound. That's why we are calling it as C2 cycle. Okay. Now this phosphate molecule is there. This phosphate is getting released here. 2 inorganic phosphate is released. Now this phosphoglycolate is converted into glycolate. 2 molecules of glycolate. It is also a 2 carbon compound. This 2 process is taking place in chloroplast. Now this glycolate is getting transferred to peroxisome, which is a single membrane organelle. So this glycolate here, oxygen is getting added and this is converted into glyoxylate. and this peroxide molecule is getting formed. Okay. Now, 2 ammonia molecule is getting added in glyoxylate, and this glyoxylate is getting converted into glycine. This glycine is the simplest amino acid. So the question can be asked is where the glycine is being formed. Glycine is formed in peroxisome. This you have to remember. Glycine is formed in peroxisome. Now this glycine will get transferred to mitochondria. Here this one ammonia molecule will be released, one carbon dioxide will be released, and from this NAD will form this NADH. It is an energy molecule. NADH is being formed. Now this glycine is getting converted into serine, another amino acid. So if it is being asked like serine is formed in which organelle during photorespiration, so it is mitochondria. One amino acid is formed in peroxisome, which is glycine. Second is formed in mitochondria, which is serine. Now, this serine is a 3-carbon compound. Okay. So, here till now, till glycine, 2 molecules of glycine was there. So, 4 carbons were there. 1 carbon get released. Now, the 3-carbon compound serine is formed. And this serine will get transferred to peroxisome again. And from this, 1 ammonia molecule will release. Here, 2 ammonia molecule were added. And now this 1 ammonia molecule was released from glycine to form serine. And now serine is releasing one more ammonia molecule to form hydroxypyruvate. And here, whatever the energy is produced in mitochondria in the form of NADH, this NADH is again utilized to form glycerate in peroxisome. So that's why in the previous slide we told that there is no net energy produced. Whatever the energy is being produced here in mitochondria is getting utilized here. So zero energy produced, net energy. Okay. This glycerate is getting transferred to chloroplast, and here and ATP is being required. So it is not producing any energy, and again it is using the produced energy which was formed in light reaction. That's why we are calling it as wasteful process. The energy which is already produced by in light reaction, it is using that, and not all it is also not producing any new energy. So and this glycerate is getting converted into PGA. This PGA will go into C3 cycle. So here no net energy is produced, and that too the produced energy is also getting utilized. Okay. The light dependent uptake of O2. Here the uptake is taking place, and release of CO2. Fixed CO2 is also getting released. See. So it is kind of red flag. Okay. This photorespiration, because uptake of O2 is taking place, release of CO2 already fixed how the plant is fixing carbon dioxide using some energy. That fixed carbon also it is releasing. Okay. And then whatever the energy produced in that cycle, it is utilizing that energy, and again extra energy is also getting utilized. So that's why we are calling it as wasteful process. So light-dependent uptake of O2 and release of CO2 in green cells of C3 plant are called photorespiration. This process creates an important difference between C3 and C4 plant. That's why we tell that C4 plant is have more photosynthetic efficiency, because in C4 plant this photorespiration does not occur. And this photorespiration occurs in chloroplast, peroxisome, and mitochondria. You have to remember which organelle is being used in this photorespiration process. Next, Rubisco is characterized by the fact that its active site can bind both CO2 and O2. This binding is competitive. It is the relative concentration of O2 and CO2 that determines which of the two will bind to the enzyme. If the CO2 concentration is more in cell, then the carboxylase part will be active. If the oxygen concentration is more, then the oxygenase part will be active. Usually, the Rubisco has a much greater affinity for CO2 than O2. This you have to remember. The affinity of CO2 for Rubisco is more. Condition for photorespiration is high light intensity, high concentration of O2, low concentration of CO2, and high temperature. Okay. During high light intensity and high temperature only this will take place. In the photorespiratory pathway, there is neither synthesis of sugar, nothing like glucose is not forming here, nor of ATP. ATP is also not forming, and no NADPH2 is forming here. Rather, it results in the release of CO2. Here, CO2 is also getting released, and one ATP is released, which is formed in light reaction. The biological function of photorespiration is not known yet. What is the reason for this photorespiration in plants, in C3 plants? It is not yet discovered what is the function for it, why it is there in plants. So we do not know yet. Next, ready for questions? Now, uh, which organelle out of these does not participate in photorespiration? We just studied. Okay. So first, this organelle which is there is chloroplast, then peroxisome, then mitochondria. So these three are there: peroxisome, mitochondria, chloroplast. So which is not involved in this Golgi body? These kind of question will be asked to you. The product of light reaction. We studied the product is ATP, NADPH2, and O2. These three are the products. So let's see, an ATP plus NADPH2 correct. NADPH2 plus glucose, this glucose is not there. Only ATP, no, this is correct. This is incorrect. And O2 and glucose. Glucose is not formed. So this is not correct. This is not correct. This is not correct. Correct option is A option. ATP and NADPH2. Next comes the photooxidation of water and photosynthesis is associated with. Photooxidation of water. Photooxidation means this here photolysis. Where it is taking place? Photolysis of water is taking place near photosystem or pigment system 2. Because we studied here the pigment system is there. This is how it is. And here this oxygen evolving complex is there. This H2 is getting splitted into H plus electron plus O2. So photolysis is taking place near PS2. So third option is correct. Next, connecting link between light phase and dark phase. What is the connecting link? Only ATP, only NADPH2, only NADH plus H, or ATP plus NADPH2. So the correct option is D, because the products of light reaction which is involved in dark reaction is ATP plus NADPH2. This is the energy which is required in dark phase. So I hope you have understand till now. So thank you for listening to the lecture. And before watching this lecture, I will recommend I'll advise you to watch the previous three lectures. Okay, so you will get a better idea of the whatever I taught in today's class. So I hope you are understanding what I'm telling. So if you if you have any doubts, so do comment. And I'll try to solve your doubt. So thank you for watching and keep watching to the Agri Addict YouTube channel. Thank you.